Nutrient cycles, I want to talk about the investigations of decay. This is also heavily on triple sciences. I do apologize for combined sciences, but this is also heavy on triple. Decomposition, something you need to know. It's a breakdown of large organic molecules to smaller molecules. So we did talk about this briefly earlier in the photosynthesis and respiration topic, but I'm just going to go in a bit more detail now that we are going through nutrient cycles. So decomposition, so usually happens when a organism dies and they're decomposed. So they're being broken down. And what exactly on a molecular level is happening is that they're breaking it from a large molecule to a small molecule. And this is carried out by things like bacteria, fungi, or decomposers. There are multiple factors that affect this. So there's temperature, oxygen, and water availability. All of these, the more that the increasing in temperature, the more oxygen, the more water there is, the increase the rate of decay of these biological material, because the bacteria and fungi are going to be able to grow a lot quicker with all of these things. So to break it down a little bit more in detail, the increase in temperature allows there to be more kinetic energy, and therefore there's going to be going to be a higher rate of decay. Similar to any other process, if the temperature is too high, enzymes are going to denature and then it's no longer going to go to work. Water availability, if there is a increase in water availability, there's going to be a higher rate in decay because water is required for decomposers, the digestive enzymes to function. You need water for these enzymes to work. Therefore, more water is going to be better for decay. Oxygen, it's good generally, but this one's be careful with this because it's only good for aerobic organisms and they inhibit anaerobic organisms. So if they're anaerobic organisms, meaning they don't rely on oxygen, a lot of oxygen is not going to be useful for them. I feel that feels very, like in the name. Anaerobic if there's oxygen, is not going to be helpful. It's the opposite. If it's anaerobic and there's no oxygen, obviously that's going to be better for those. The rate and change of decay is just change in value over time. You might get a math question on that. And with gardeners and farmers who are trying to make these decomposition happen, usually they find the optimum conditions for very, very quick decay. So a good temperature that's fast enough but won't denature, enough water, and enough oxygen. Because they want this decay to allow for these biological material to produce compost. And compost is a very, very useful material for gardeners and farmers to use. Anaerobic decay, again, I said it does not use oxygen, and this produces methane gas. And this methane gas can be carried out in, this production can be carried out in a biogas generator, and this allows it to produce fuel. This is something, good to know. The most common practical that they use for investigation of decay is the milk experiment, and is when the milk decays. This is very important to know, actually. When milk decays, milk has lipids in it, so fats is one way to explain it. These lipids are going to break down into glycerol and fatty acids. And because fatty acids they're acids, they're going to make the pH very low because they're very acidic. And the way this experiment works is that they monitor the pH to figure out how fast the milk is decaying, because the faster the milk decays, the more it's going to turn into glycerol and fatty acids. The more fatty acids there are, the lower the pH. So they usually they set this up with different temperatures, different pHs, and they do it at different intervals. So we're going to look at this experiment right now with one of these questions. So here, this is a math question. Again, look through this. We're not going to go through this. On 4.5, it tells you milk contains lipids, and lipids are broken down when milk decays. And it's asking you to explain why the pH changes more quickly when the temperature is higher. We know that as temperature increases, the rate of decay increases because there's more kinetic energy. If the rate of decay increases, the lipids are going to be broken down more quickly, and therefore more fatty acids are produced. Therefore, the pH is going to change much more quickly, and the pH is going to drop down so much more quickly because there's more fatty acids being produced. I hope what I've said is clear. Here are the answers. Enzymes are more active, more bacteria produced, lipids are broken down more quickly, and therefore fatty acids produced more quickly. Any questions on everything we've done so far? Any questions? Any questions on that? Let's hope. Okay, can you explain control variables, independent and dependent? Yes. Control variables is what you want to make sure it's fixed, so you make the thing fair. So if I were trying to figure out how temperature affects let's say affects respiration, not respiration, let's not do respiration. Let's do that. How temperature affects respiration, or not photosynthesis. Let's do photosynthesis. How temperature affects photosynthesis. Sorry. The control variables would be anything but basically temperature, because the independent variable, in this case, the independent variable, which is the thing you're investigating, is the temperature. That's the independent variable. Control variables are things you don't want to change. Things like the number of plants, the type of plants, the light intensity in the room, the oxygen, any variable that could affect the experiment, control it to make sure it stays the same for everything. Anything but the independent variable to me is control. Dependent variable is what you're hoping to find. It's like your hypothesis. So in this case, the dependent variable would be you think that an increase in temperature is going to allow for a higher rate of photosynthesis. Is that